Our next speaker now is Dr. Tiffany Vora. She's the VP of Innovation Partnerships at Explore Mars. She's also a Singularity Fellow in Biotechnology at the Singularity University. Ready to go? Okay. Thank you very much, Tiffany. Welcome to the stage.
Thank you. Good morning, everyone. Delighted to see you again. Thank you to those of you who came out last night for our panel, which went almost twice as long as I was expecting. So I think that's the sign of a good conversation. But today, for something entirely different, I'm going to be pulling further on the biology thread right now and talk to you about harnessing synthetic biology on Mars. We're very fortunate here at this year's Mars Society that we have quite a few folks here who are doing this stuff with their own hands. And the next speaker after me, Dr. Erica, she's going to be telling you even more about the work that she's doing. So my job here, Erica, is to set you up. Okay, so imagine I told you about this magical manufacturing technology that is self-perpetuating and self-repairing. It's highly modular. It's flexible in terms of the input that it takes. It is programmable and reprogrammable. It's resilient, and it's remarkably stable, that if you keep it cold and dry, it'll keep running and work and wait around for you for years to decades to centuries to millennia to perhaps even more than that. Now, the good news, of course, is we already have that technology. That technology is life. And here on Earth, we've had life for the last 4 billion years, having these properties as this type of system. And so at Explore Mars, we are very interested in the various technology spaces and innovation spaces that are not only pertinent to life off Earth, but also to life on Earth today. Now, I am a molecular biologist by training. I am very interested in the biotechnologies and everything that they touch, which of course in my mind means space. I'm highly biased. But so what we decided to do a couple of years ago at Explore Mars was to spearhead a synthetic biology working group to take a deeper look at what we would actually need in order to further biotechnology and synthetic biology specifically for space, but also with a mandate for making life better on Earth at the same time. These are the members of our working group, and I'm going to be talking today about the findings of our working group. I mentioned Dr. Erica De Benedictis. She's your next speaker, so I will be talking very lightly about the stuff that she does. We had Dr. Lynn Rothschild from NASA Ames and Brown University, who is one of the pioneers of this field, and Dr. Niels Averish, who is currently at the University of Florida, who's been doing really interesting work with synthetic biology for space, particularly around biopolymers and plastics. So here's why we're so interested in this space today. It's because we're at this almost magical moment in history where, thanks to technological innovation, the boundaries between the living and the non-living world are breaking. One of the things I'm going to encourage you to do starting today, if you haven't already, is to not think of biology as separate from technology, but rather to think of biology as a technology. And as our exponential technologies continue to become cheaper, faster, more powerful, more accurate, so will our biotechnologies. They're working on the same types of curves. Now, in terms of synthetic biology, biomanufacturing, and biotechnology, we're seeing a push from governments around the world to build up and beef up their bioeconomies. There's lots of reasons for this in terms of things like sustainability, resilience, supply chain resilience in particular, circular ecosystems, all of these things. We've seen really strong plans come out of countries like the US and also, of course, the EU, but lots of other nations around the world, including space-faring nations, have also been working on their bioeconomy plans for in the coming years. We're talking, of course, about countries and organizations like the UK, the UAE, the Saudi government, China, Germany, all have very strong bioeconomy plans. And this is more than just thinking about recycling or more than just biofuel. This is about, again, using biology as a technology in order to make all the stuff that we need for our lives. So there are lots of strong benefits to what I think of as a circular bioeconomy, and I think of it in 3 big buckets. Like, yes, we're talking about protecting the environment, but we're also talking about building our bioeconomies in order to support socioeconomic development and to support industry, right? We're talking about real prosperity here. We're talking about jobs. We're talking about new products and services. And many of these feed very nicely into the UN Sustainable Development Goals, in case that's something that you find exciting. Now, what I always tell people is this is the vision that we have for life here on Earth, But also, of course, this is a vision that we could have for anywhere that humans call home, whether it's low Earth orbit, the moon, Mars, or even further beyond. So when you think about biotech in space, you're probably thinking about things like these bioregenerative life support systems, which we've seen in play for many, many years now. These are where you've got these closed series of nested closed loops that enable you to use biotechnology and biology to do things like mitigate waste, produce food, keep your air and your water clean, grow your food, that kind of thing. And so I want us to focus today, though, on this component, the microbes. And Stephen set us up really nicely for this. You guys are going to have bacteria on the brain by the time you get to the break. And I, of course, couldn't be more excited about that. So let's focus in a bit more on this idea of what bacteria can do for us in space. Now, they're here as part of this kind of integrated look at microbes in human health and how microbes might be impacting human health in space. Now, of course, for many, many years, the basic idea was microbes in space, bad, right? They make biofilms, they foul up our tubes, they make us sick, they do all these things. But fortunately, we've come to a much more nuanced understanding of how important bacteria are not just to human health, but also to all the stuff that we want to do in these extreme environments. And so I'm going to focus on this bubble right here, these bioengineered microbes, and how we can leverage them in conjunction with our biotechnologies in order to make the things we need. Now, our Explore Mars SynBio working group, we went through the 2020 NASA technology taxonomy. And of course, being all biologists, you were like, oh, bio is good for everything. We're like, no, let's focus a little bit more carefully. And we ended up zooming in on these 3 technology areas that we thought were probably places where the affordances of biotechnology were going to be really strong. Why you should look at using biotic rather than abiotic approaches to solving these problems. And I'm showing you the 3 technology areas here. These are exploration and destination systems, which of course includes in-situ resource utilization. We'll come back to that idea. Human health, life support, and habitation systems. And then on the right, material structures, mechanical systems, and manufacturing. And the reason we focused in on these 3 taxonomy areas was because we came up with these sort of 2 big ideas, these 2 big themes about why you would want to use biology for these things. And the first one of these affordances of using these biological systems is it takes you from a take paradigm to a make paradigm. We believe that biology is going to be very powerful for doing things like solving up mass constraints, because again, biology will make more of itself. All you have to do is fit the pieces that it wants and a bit of energy, something to eat. And it'll also make the things that you want it to make more or less. The biology is complicated. That's why we're biologists, because it's exciting. But that enables you to go again from take to make, which will be really, really helpful when we're talking about these excruciatingly long, excruciatingly expensive supply chains. And that, of course, allows you to go from a stored mentality to a fresh mentality, whether you're talking about food, whether you're talking about materials, whether you're talking about medicines, therapeutics, that kind of thing. Being able to get a small amount of exactly what you need, exactly when you need it, is something that we think biology is particularly well suited to. So now, yesterday, Melody Yashar gave another one of her great talks talking about building on Mars. And she talked a lot about using regolith as a building material. Love it, of course. But Lynn from our working group has also been doing things like working on mycelium biocomposites as a construction material that you could use for Martian habitats. Mycelium being the furry stuff that yeast throw when you make them really, really happy, or mushrooms do. And actually, yesterday on my way to this conference, I stopped off at the BMW Design Laboratory and got to handle a bunch of their mycelium-based leather, right? No cows, just fungus. And it was pretty cool, actually. Some really cool stuff. So we're seeing lots of activity in this area about using bio-based materials for a variety of applications. And we would love to see us thinking more about that for space and for Mars. But what I really want to talk about here is synthetic biology, right? So synthetic biology is when we bring together things like genetics, computer science, physics, a bunch of other types of scientific disciplines in order to program or reprogram living things to either make new things or have new functions that they didn't have before, or to improve the functions that biology has already given us. And so the way I want you to think about this, just briefly, is to think about DNA like a source code. In Stephen's last talk, he was talking about RNA, which you can kind of think of as compiled code. But if you want to use that computer analogy, you can also, since I mentioned cars, think of DNA like the chassis of a car that you can add things onto or take things away from in order to change the function. So what we can do with DNA code is we can add modules into the genome of an organism in order to make something new, to improve a function or a molecule that's already there, or even to do something entirely new, like light up if you want your bacterium to act as a sensor, or to move into a particular place. All of that becomes possible using this synthetic biology paradigm. So again, biology as a technology, as a programmable technology. So where do all these DNA modules come from? The good news is everywhere. Biology has been innovating on Earth for 4 billion years, and that means if we look at the whole kingdom of life, which is up here, we're able now, thanks to super cheap, super fast DNA sequencing, to grab these modules that evolution has already worked on all through the kingdoms of life, right? Whether we're talking about the bacteria that live in your gut, the bacteria that we've been using for 10,000 years to make yogurt, synthetic bacteria that do all of this. And, by the way, this kingdom of life that I'm showing you here, all of this stuff, all at the top, that's all single-celled life, okay? All of the multicellular life, all of the mushrooms and Galapagos tortoises and like me and my kid, we're all the way down here in a very, very tiny corner of the kingdom of life. So we have a ton of inspiration to look to from the natural world in various environments and various forms of life in order to grab these DNA components to try to remanipulate them. But wait, there's more. You might remember that last year's Nobel Prize in Chemistry went to 3 gentlemen who've all been using AI in biology. I love ChatGPT as much as the next girl, don't get me wrong, but I'm telling you that the killer app for AI is biology. And the folks from Google DeepMind who won last year's Nobel Prize had worked on a series of algorithms called the AlphaFold algorithms, which first were able to solve to predict a protein's structure from its function. Back when I was a graduate student, which wasn't that long ago, thank you very much, it could take you 8 years in order to do that. These folks released a database with structural predictions for every protein known to man, 200 million of them several years ago. It's a big deal. They've also had other algorithms that will do things like predict 3-dimensional molecule structures and how they fit together. They also have one called Alpha Proteo, which makes predictions for protein structures that, as far as we know, have never actually been tried on Earth before. And a new version from DeepMind is this Alpha Genome, which is really quite remarkable. It's an algorithm that unifies a whole bunch of types of biological data, again, to make predictions about if I change this in the DNA, this is what I would expect to happen. It's incredible. And remember, Google DeepMind is not the only company working in this space. There are many, 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 many innovators out there looking to harness this AI revolution for biology. A simple example here is an algorithm that came out of a collaboration between NVIDIA and Stanford, which this is essentially an LLM for writing entire DNA genomes. Now, to my knowledge, none of these completely artificial genomes have been booted up into a living thing. If you know differently, please come find me during the break. But it's a matter of time as we start using these brand new tools to see which ones are actually useful, which ones can we use in order to expand the repertoire of things that become possible when we look beyond what, to our knowledge, has happened thanks to biological evolution here on Earth. So hopefully what I've convinced you in the last 15 minutes is that synthetic biology is awesome, right? So your question should be, what's the holdup, Tiffany? Like, why are we not doing this right now for our Mars missions? So let's take a look a bit more about what's actually needed. So any mission is going to have a particular profile of stuff that it takes with us. Right? Any mission class, whether it's chemicals, whether it's packing material, any of those things are going to all be available as the inputs to a synbio system as well as what other in situ resources we have available to us based on where our mission is actually going. And so you've got to take those inputs into consideration when you're figuring out what synthetic biology tools could we be designing and using actually while we're there on the ground, or if we send these missions ahead to prepare for humans coming. Niels has done a lot of really excellent work on this. I'd like to point you toward his papers. I'll be giving you the references down here in the corner, or you can always email me and I'll send you the papers. But there's it's an interesting chicken and egg problem, right? In order to know what kind of tools we can build for the mission, we need to know what the mission looks like, but we still don't know what the missions look like, right? Like, how much methane are we going to have? How much, we don't know. We don't know these things yet. So we get caught in these loops, but there's some really interesting work that's been done on thinking about how to solve that problem. We also really, really need to mature our current biotechnologies in order to make them useful for these near closed or preferably fully closed loops. Sorry about that. And so here again, I want to zoom in for just a minute on the food and medicine production part of this. We think biology is useful for all of these, but when we again think about food and medicine, I feel like that's where biology really shines here. So Lynn has been working on this idea of an astropharmacy. We don't know what's going to happen to our crews over their 2+ year mission days. So what if instead of having to take every single drug that you could possibly need, oh, and by the way, they may or may not be shelf stable on the way out there, what if you could make, again, exactly what you needed using a very, very small amount of mass that you bring with you in order to meet all of those potential health, or as many of those potential health situations as possible. And so imagine instead of taking a huge library of these things, you're able to take some bacteria, a bacterial library with you that has been programmed in order to make biological and peptide-based therapeutics, and you wake the bacillus up when you need them, and they go ahead and they make in a small batch on demand exactly the therapeutic that you need, right? So as Lynn says, you could have a library of thousands and thousands of kinds of drugs engineered into different cells. And we're talking about, not even grams of mass potentially that you would need to take with you for the bacteria themselves. So our working group identified 3 places where we really think technological process and research and development needs to be accelerated in order to bring us to this point. The first has to do with getting shelf-stable reagents. We would really prefer to see things not need to be refrigerated, not need to be frozen, not have to worry about the cold chain. That benefits Earth as well as space, so that would be something we would love to see. We also need to see flight-tested hardware, the bioreactors for actually building, growing, maintaining these bacteria, and then processing what they make in order to purify out what you need. The flight-tested bit is the solvable problem, but it hasn't been solved to our satisfaction yet. And then the last piece that we identified as being ripe for innovation is looking at the chassis organisms, the biological entities themselves that would actually be running this. And there's, as the young man in the front pointed out, there are things we can build on here from evolution on Earth, and that we're really keen on that. There's a whole bunch of considerations about how you choose which microbe you want as your chassis. I am actually not going to talk about that today. I am going to set Erica up to talk about the amazing work that her group, Pioneer Labs, has been doing for this exact problem. So you get to hear it straight from the horse's mouth instead of from me. So stay tuned for that, please. But what I want to point out here is there's a suite of these technologies here where additional research and development, and specifically dollars, as we talked about a few minutes ago, are needed in order to mature the technologies to advance their TRLs to get it to a point where we could actually use them, right? And on the left, you can see we've got a bunch of applications here from plastic and consumable recycling, bioremediation, waste recycling, e-waste recycling, regolith remediation, and then as we talked about, those structural components that you could be making with your biomaterials, pharmaceuticals, mining. I mean, there's all kinds of applications here. Some of them are closer to prime time than others, as you can see, and so there's always more work to be done. And I would argue that underlying all of this, what we really need is a strong and powerful and predictive digital infrastructure that enables and is going to empower us to not just do everything ahead of time and bring it with us, but to empower our crews while they're there on Mars in order to solve the problems that come up in front of them when they need them to be solved. And again, biology is a spectacularly nimble system, and it would be a shame to put that affordance down. Also, I really kind of like the idea of the crews having agency on this from a human factors perspective. If you've ever lived in an extreme environment, I was MDRS 3 and 11 and FMARS 10, so I remember I was in Antarctica not too long ago. Actually getting to do the thing that makes your life better is huge for you and for your crew as a team. And so I would love to see us leveraging biology to give us that human factors boost as well. Okay, so if you go and you look at our paper, which we published last year in New Space, you can get a bit of a deeper dive to see how we see that pursuing bioeconomy and synthetic biology on Earth radically empowers what we can manage to make happen in space, on Mars in particular, and vice versa. So last night you heard me talk about how important it is for us to emphasize that innovating for space makes life on Earth better. And I think that synthetic biology is a really fantastic paradigm of that. So please feel free to go deeper there. So the overall vision that I'm selling you here is a world in which biological systems, our 3D printers, our computers, our sensors, our power plants that help us make all the food, fuel, and fiber that we need in order to live resiliently, sustainably, and to thrive and not just survive in the places where we are, right? And that's anywhere that we call home. I'm going to pause here because I reckon there's probably some things you guys want to talk about before we move on. So this is me. This is how we can stay in touch. Feel free to reach out to me on LinkedIn. If you do that, please let me know that we met here. There are a lot of random people on LinkedIn, and I don't want to talk to them. I want to talk to you. This is just kind of the beginning of what's possible here. And I love this. I mean, I'm a biologist. I'm highly biased, but today is the most boring biotech and synthetic biology are ever going to be. It's only going to get better from here. Thank you very much. Let us talk.